Okay friends, now we're going to talk about the RNA or ribonucleic acid. It is the second most common important ingredient among those nucleic acid family that we know. That DNA is the boss of RNA. If we consider DNA is the boss, because DNA is having all the blueprint of life, all the informations, but not only they are having the information on their own, but they cannot do anything except for their messenger, which are RNAs. So RNAs are playing a very important role inside cell to convey the message of DNA from DNA to make protein. So what we have to do, we have to make proteins, because these proteins are everything, almost everything we are looking at as life. Now we have DNA as a messenger, and we have everything written on this DNA. Now DNA cannot produce protein on their own directly. Okay, so it needs some messenger to carry this message from DNA to protein, because if DNA goes for that, according to the evolutionist, if DNA goes for that, then DNA can be prone to damages, and if any damage happen to DNA, that will reflect in the next generations. That's why in this type of central dogma of biology, the DNA has been restricted for directly go and go and involve in the process of translation. So that's why they produce RNA from DNA, then this RNA goes and finally goes for the protein synthesis. Okay, so that's why we need the presence of RNA in all these cases. Now let us first talk about the functions of RNA in cell. Now first, we know there are not one type of RNA we can find in the cell. Most of the times, generally three types of RNA can be found. One is called the mRNA or messenger RNA. Second one is called the tRNA or transfer RNA. And third one is called rRNA or ribosomal RNA. Now this mRNA is the actual messenger for the DNA code. So DNA have the code sequence, and mRNA converts those sequence as code, and they take them as codes, and they helps this code to organize different amino acid sequences during protein synthesis, and thus a particular protein can be synthesized regarding to a particular code or being directed by a particular code. And this transfer RNA helps to carry or helps to transfer amino acid sequences from here to the polypeptide growing chain. So we have a growing chain of polypeptide during the protein synthesis, and transfer RNA actually carries the specific amino acid according to the code or obeying the code which is present in mRNA. Okay, so these two RNAs are interacting during the translation process. Now what is the third kind, or rRNA? As the name suggests, it means the ribosomal RNA. So this is the structural component of ribosome. So what is ribosome? Ribosome is the machinery or workbench for protein synthesis inside a cell. So we have a cell, inside that we have ribosomes, and that ribosomes are made up with these rRNAs and several different types of proteins. Now they organize themselves to make this ribosome. So what happens during protein synthesis? We have this mRNA here, this is the mRNA, and we have ribosome sit on there, we have the ribosome here, and we have this tRNAs just stand here like this with those codon and anticodon loops, and this is the tRNA and rRNA present in the ribosome. So that's how these three types of RNAs are interacting during the protein synthesis, and it leads to the formation of protein, which is really important. Now if we look for the function, as I have told before, the functions of all these different types of RNAs, but still the functions is normally as an adapter molecule, as I have told before, because this is the adapter of the information from DNA to protein. Okay, now RNA can also play the structural role, as you can see in case of rRNA, which helps to make the structure of ribosome along with different types of protein. Now RNA is a regulator molecules which through sequence complementarity bind to the interface with the translation and certain mRNAs. So we have seen in this case. So the main important goal of RNA is to carry out the translation process, and to carry out and to take the message from the from DNA and take it inside the cell, into the cytoplasm, and finally go for that. So if we look for the eukaryotic cell, In case of eukaryotic cell, we have the cell. Inside the nucleus, we have the DNAs. So DNA cannot be cannot come from this nucleus to the cytoplasm, because there are different types of enzymes that can easily degrade DNA. So DNA is being put in the separate area like nucleus. So what they have to do, they have to produce. They need some adapter. They need some carrier of message. So the message carrier will carry the message from DNA and they can go to cytoplasm, which are not prone to damage. And this role is being played by these RNAs, especially mRNAs. Okay, and tRNA is also being made in such a way. RNA is also being made such a way. When they come into the cytoplasm, they organize themselves. They interact themselves in this way to make proteins. Okay. So that's why it's important. And another more important factor, which is denoted few years back, that is the RNA can act as enzymes. We call them ribozymes, because the RNA is according to the chemical structure of RNA. If we look at the chemical structure of RNA, actually chemical structure of sugar that is present in RNA. So the only difference between a DNA RNA chemically is in the backbone is that varying in the sugar or type. So like that in DNA we have the deoxy sugar. It's also ribose sugar, but it's deoxy. That means in second carbon position we have only hydrogen in case of DNA. But in case of RNA, instead of having only hydrogen, we are having hydroxyl. So instead of presence of this hydroxyl, as a result of the presence of this hydroxyl, it can act like a catalyst. So it can cleave the itself from the backbone via cross-linking the phosphate groups, and as a result we can have the hydrolysis of RNA really easy. Okay. Now if we look for the structural detail of RNA, what we are having the sugar, as we know, but this sugar is a ribose sugar, not a deoxyribose, because it's 2 carbon position. We have the hydroxyl group instead of hydrogen, which we can find in case of DNA. Now a phosphate group has to present there for attaching or for combining those via the phosphodiester linkage. So we have a phosphate group, and we have 4 types of bases. Now here are another difference of RNA with DNA, because among those 4 types of bases, in DNA we are having adenine, guanine, thymine and cytosine, but in case of RNA we are having uracil instead of thymine. Okay, but we can see now that the sequence or the structure of uracil is very much similar with thymine. So that's why this uracil is replaced with thymine in case of RNAs, more especially this uracil. Why they have this uracil? We do not have the answer. Actually, we do not have the proper answer, but we know that if this uracil present, that uracil can have a remarkable ability to bond with different molecules, different bases like guanine. So guanine can in turn pair with uracil, and this kind of pairing gives a structural rigidity, a structural stability to the RNA sequence or to the RNA structure. That's why might be this uracil is present instead of thymine in case. Okay, so these things are assembling like the way we have seen in assembly of DNA. So this is the backbone portion, and this is the base popping out of it. So that's will be the structure. This is the base, and that's the backbone is creating. So here is the glycosidic linkage, here is the phosphodiester bond, and another phosphodiester bond will be here like that to attach with the next nucleotide. Okay. Now let us look another important feature of RNAs is the presence of some modified nucleotides. So as we know, normal situation we have adenine, we have guanine, we have uracil, we have cytosine. These 4 types of base are being found inside the RNA structure. But we can also find several types of modified nucleotides like pseudouridine. Now you can see, if you look at the structure of uridine, pseudouridine is resembling the structure of uridine, but instead of it being an uridine, it is having slightly different structure. That's why we call them the pseudouridine. Okay. Now it can also produce the dihydrouridine. As you can see, these places of uridine has to have 1 hydrogen in nature, but you can see this place and this place, this 2 carbon position, we have 2 hydrogens instead of 1 hydrogen. That's why we call them the dihydrouridine instead of normal uridine, and we denote them with D. In case of pseudouridine, we denote them with psi. And we also have nucleotide bases like inosine. Now what is the important feature of this inosine is this inosine molecule resembling the structure of all different types of bases in such a way that it can bond with other type of bases or any other type of bases. So whatever we are having, we normally know that A always pair with T, but in this case A always pairs with U, because instead of T we are having U. G always pairs with C. That's a basic rule, that's a Watson-Crick rule. That's our Chargaff's ratio rule. But the problem is, in this case, when the inosine present, then the inosine have a ability to bind with all of these 4 types of bases. So inosine can bind with adenine, it can bind with guanine, guanine, uracil, and cytosine. So it has a remarkable ability to bind with or make a bond with all of these different types of or nucleotides. Okay, so that gives them this RNA molecules a remarkable way of binding itself, or remarkable way of stabilizing the structure of itself. Okay, so that's why this presence of this inosine can change the stability of RNA, and also it can change the stability of RNA. It can change the RNA of making, it can increase the ability of RNA to make second. Okay, now let us talk about the functions of modified bases. As we can see, we are not only having the bases in RNAs, but we are having slightly modified bases. Instead of having uridine, we are having pseudo-uridine. Instead of having uridine, we have dihydrouridine, which is actually changes in different places of that base ring. So as a result of these modified bases, most of the time RNA structure is being favored. And what is the importance of this structure? Now let us consider. Now, some of the known functions of modified bases are to confer the increased stability on RNAs, because you can see, and we will going to see in case of the tertiary structure of RNA, as we can see in case of mostly the tRNA. In this tRNA, we can see the characteristic L shape of the tRNA, and that L shape of the tRNA is being only been made by using the modified base, by using bases like pseudouridine, dihydrouridine, or something like that. Now, to modulate their recognition by proteins to other RNAs and translation apparatus, because you can see the normal situation, what happens when we need some protein molecules to be attached with those RNAs to start something like translation, to start translation, or to begin different reactions. In those situations, the presence of few modified bases actually tag the position for the protein binding. So what happens if we have a stretches of RNA here? For example, here is the RNA, and this region contains the modified bases. So that means in this whole RNA, for example, these 2 regions having the modified bases, for this whole RNA, this region suggests the protein to bind with this region of the RNA. So it suggests this protein to bind with this region. So that is the work of this modified bases that we can find in the cell. Okay. Now, roles for modified bases in recognition by amino acyl tRNA synthetases. For example, it has been clearly defined in a number of cases. As you can see, the roles of modified bases also the recognition by amino acyl tRNA. Now, what is amino acyl tRNA? As we can know, amino acyl tRNA is a tRNA, or transfer RNA, which carries amino acid with that. So we have a tRNA. Here is the anticodon region with 3 sequences, and here we have the amino acid linked to it. So what happens? This amino acyl tRNA contains if we are having this mRNA having the slightly modified bases, then this amino acyl tRNA can recognize this part of the mRNA very quickly. So that's why the presence of this kind of base pairs, or this kind of unusual bases, are important, or modified bases are really important in few situations. Now, what are some of the unique base pairs found only in RNA? As we can see. Okay, let's first talk about the unique base pair, which we know in this case. Look at the GU base pair. Now, as we know, in normal situations, G always pairs with C and A always pairs with T, using the Chargaff's ratio, and normally this GC pairing is been done using 3 hydrogen bonds, and AT pairing has been done by 2 hydrogen bonds. But we can see in case of RNAs, exactly G can pair with U, because the uracil having just slight structural variation from thymine. Though it is very similar to thymine, but still it has some structural differences, and those structural differences actually help this to bind with this guanine. So guanine can in turn bind with uracil using 2 hydrogen bonds. As you can see, one is here and another hydrogen bond is here. Okay, so these 2 regions. Now, when they linked together, as you can see, when they link together, that gives some structural a dynamistic property to this RNA. That's why this kind of bonding is being favored. Okay, so what happens, as we can see in this picture, it has been studied that this kind of bond, this kind of GU interaction in the inside the RNA sequence increase the capacity for self complementarity, because as we know, this RNA is having a strand. We have some 5' region, we have a 3' region, and in between there we have base just popping outside the environment. So it cannot be established, it cannot remain like this, because these bases are vulnerable to damage in this, if it remains like this. These bases are vulnerable for the hydrophilic attack in this portion. So that's why this base always try to make hydrogen bonds with each other by coiling themselves up, like we can find in DNA. DNA is much more stable, that's why. So RNA is much more fragile. So that's why RNA need to fold on their own. So what happens, it will fold, make some structure like that, and this kind of structure you can see there are hydrogen bonds in few regions, and in few regions there are no hydrogen bonds. Few regions there are only bases which are popping out. So this kind of structure is called secondary structure, and further folding of this kind of secondary structure will create the tertiary structure or third degree structure for an RNA molecule. Now if we are having this GU pairing along with AU GC or GC pairing, then it actually increases the freedom of making base pairing among the self strands, among the self bases of this RNA, which actually help to increase the capacity of self complementarity in the RNA molecule. Okay, so that's why what we are doing by putting this U, and by the presence of this U bonding, it increases the capacity for self complementarity, and it increases this RNAs to, it actually help the RNAs to fold on their own to make secondary and tertiary structure pretty good. Okay, so now let us talk about the primary and secondary structure. So as we can see in this picture, we have a backbone of 5' 3' direction. This backbone is made up with phosphate. Along with that we have sugar. In this sugar we call the ribose sugar. So this is not deoxyribose, so the second carbon position of the sugar contains hydroxyl instead of hydrogen. Okay, and this sugar is bound to bases. Those bases are popping outside into the environment, as we can see in this picture: uracil, guanine, adenine, cytosine, some like that. So this base pairs, what we can see, in normal situation it is vulnerable to damage, it is vulnerable to different solutions. As we know, those are hydrophilic, and those are not good for attaching with these bases. That's why these structures need to be folded. So when this structure is starting to fold, they make different structures like that, as you can see in this picture. Now the structures, as we are seeing, these are the very much common structure that are most of the time is being found, but there are a lot more different types of structures in which an RNA molecule can adopt. Now here we are talking about the bulge loop. This bulge loop structure, you can see one strand remains something like this. We have the strand is going on like this, and here is the strand, the same strand is folding and makes something structure like that. Okay, so after folding it will create a structure bulge like that. Why this kind of bulging is happening? Because of the bases are present there. These bases are no longer able to bind with themselves, because we have a GC, but this is not in close proximity or same orientation. So G in front of U, A in front of C. So this kind of bonding is not possible in normal situation. Now we can also have seen the bonding of AC, but in case of mutations and in case of the transition from GC to AC, something like that. Okay, now secondly we talk about the internal loop. In the internal loop model, what we are looking at this parallel nature. So this part of the RNA molecule just find the bases are able to find their own partners, so they can easily pair with themselves, but few regions or some regions in between these RNA sequences, some bases are there which are unable to find their partner, which cannot make any bond. So in those regions, what we are having a gap, we are having a just bulkier region, which is called the loop, or loop like structure is being created. Now in this picture what we are looking at, multi-branch junction has been found. So this junction, we can see this is like a crossing, so 4 road crossing. So in this picture as we are looking at, these are the most of the part of these strands are actually able they are able to find their partners, and very few regions in this particular region in internal place, there are no other base pairs remain there to make bonds between themselves. So that's why this region remains bumped like this, and all these other strands start to interact with each other. They make bonds with each other, they make hydrogen bonds with each other to finally make what we call the multi-membrane junction. Okay, and we also have the stem loop structure, which is a pretty common structure. Why it is called a stem loop? Because we have a stem coming out from the soil, as you can see, and another loop here. So that's why it's called the stem loop structure. In this structure, what we can look here, this is one strand which is constant, and another strand is coming out, and few regions are joined and few regions are not, and few regions, as you can see, which are unable to join with themselves, are just living like a group. Okay, like a loop, sorry. So in all these cases, in all this secondary structure, one thing we can look for common, that this kind of structure is being formed for two regions. One is we have some regions where base find complementarity, so they can pair bonds with themselves, and there are few places inside the RNA where the bases cannot find any complementarity, so in those cases they remain like a loop. So that's why what we are having, we are having punctuated interrelation, punctuated sequence interactions via loops. So what we are having, we are having a straight interacting chain, then punctuated by a loop, then another chain, then punctuated by another loop, and that's how these structures, or the secondary structures, as you can find, the double helical structure of an RNA, which is being made due to the interaction between bases, is being punctuated by this kind of loops that are present in middle regions or middle portions. Why these loops are being found? Because in this case there are not that much presence of different base pairs, or there are no bases present which can really find their partners like GC or AT or GU kind of pairing. So that's why in these situations these loops are being formed. Okay, now if we look how the secondary structure is formed, so this is a straight line, and this line is finally folded. As you can see, in this region there are regions like GC, AU that they can pair bonds with themselves like that. But in this region we are having GG and AA. So this is a structure, these are the base sequences which no longer can form any bonds, because there is no C. And if we are having G, we need to have C to make bonds, and we are having A, but we are not having any T. So this is A and G, all purines. So all the purines cannot make any bond with themselves, so we need purine and pyrimidine to make bonds. Not only the purine and pyrimidine to make bonds, but also the right purine pyrimidine combination to make bonds. So what we can find in this kind of loop, we cannot find those interacting or complementarity purine pyrimidines. That's why we cannot find any interactions or any kind of double structure like this. Okay, so these regions, as you can also see in these loops, still this kind of formation is done. But some regions where you find the complementarity, RNA start to pair with themselves, RNA start to pair with themselves like a velcro, and to make bonds like that. Okay, and the presence of these secondary structures are really important, because we cannot store RNA in environment something like that, because bases are vulnerable to damage. That's why we cannot store them. We need to fold the structure together, and this folding of the RNA helps to for sometimes to carry out very different tasks pretty easily, because our RNA molecules have to be carried out from after the synthesis of RNA molecule inside the nucleus. It is carried out like a linear structure like this. Then when it comes to cytoplasm, it start to fold and make a structure like that. Okay, and in the process like translation, we need the presence of small molecules, not the larger molecules like these linear RNAs. Okay, so that's why these structures are important. Now if we look some of pretty unusual secondary structures are something called tetraloops and pseudonodes. So what these tetraloops and pseudonodes? So as you can see in this picture, that additionally interactions with ions, small molecules and large proteins may influence. As you can see, for example, a stem loop with the tetraloop sequences UUCG is extraordinarily stable due to the spatial base stacking interactions. Now as you can see in this picture, in this tetraloop structure, let me tell you, tetraloop is nothing but a stem loop structure. So what we are making only a stem loop structure, which in the previous picture, as you can find here, something like that. Here we have, so here we have a stem loop structure like this, and in this structures, this part we can find pairing. But what happens during this kind of pairing, if we are having more U, C and G in this situation, more importantly, two or more than two uracils and very guanines present there, then this guanine have a remarkable ability to pair with uracil as well as cytosine. So what they can do in these situations, as you can see, so this guanine pairs with U, guanine pairs with C, and as we can see, this these are the simple bonding, these are the hydrogen bonding as we know. But except for this hydrogen bonding, the presence of C, cytosine and uracil and guanine in this kind of orientation, what happens, we are having this base stacking interaction that is going on, and that is shown in this picture between these bases. Okay, so uracil, cytosine and guanine base stacking interactions, and these base stacking interactions are really strong. This base stacking interaction between the UUCG is extraordinarily stable due to the spatial base stacking interaction loop that is being formed in this kind of picture. Okay, that's why the presence of this kind of tetraloop structure is really important. Now base pairing can also take place between sequences where are not continuous with each other, as you can see in this picture. So we have a stem loop structure again like that. Now in this stem loop structure, you can see, as we know, in this stem region there are complementary sequences which can make bonds with themselves and to make a stabilized structure like that. But in the upper loop like structure, there are some regions which are popping out. There are some nucleotide bases which are popping outside. Okay, now these bases can have a complementarity in the other part of this structure of this tetraloop. So what happens, this base start to make a pair, start to make bond with this region, so with the sequences that we can find in this below structure like this, or in this ground structure like this. Okay, so what happens, this ground structure start to come up, and the base sequences we can find in the ground, we start to pair with these bases that we can find popping outside from the loop, and they can make structures like that. Now this kind of structures is being called the pseudo knot structures. Okay, so what we are having the tetraloop structures, as you can see in this picture, and we are having the pseudo knots. Now this kind of structure pretty unusual, but you can normally most of the times we cannot see this, but these kind of structures are important to make the RNA structure stable. And not only to make the RNA structure stable, but the presence of this kind of structure actually denotes the RNA, or actually flagged the RNA with some of the properties that come, they actually call proteins that come and bind to us because we are having this kind of regions. So that is this part of the region of the protein, this is of the RNA, actually act as the flagging instruction points. Okay, now if we think about the conformation of RNA double strand, so when we talk about this kind of strands where we can find the double strand interaction of RNA in between RNAs, then we can find the interaction. So what kind of RNA is going to make, what kind of direction this helix is going to propagate when the two strands are coming together to make a double helical RNA. Now according to the presence of, as there is a presence of the 2' hydroxyl group in the ribose sugar, it is conformationally flexible region of an RNA molecule, and as we know, the presence of this 2' hydroxyl molecule in RNA prevents this RNA double helix to maintain the B form. So instead of maintaining the B form, it helps, or it triggers this RNA duplex to form the A form of the RNA, to be the A form, or to make an A form of the RNA. Okay, so what do you mean by this A form? As you can know, this A form is slightly bit more zipped than the B form, and it is also very it is not stretched, it is zipped, and minor grooves are narrow and deep. Minor grooves are not narrow, minor grooves are deep, and major grooves are narrow in this case. So it is making the A form stable because of the presence of the hydroxyl 2' hydroxyl group in the ribose sugar. Okay, Now if we look at the tertiary structure of RNA, what we can find, the RNA can not only adopt the secondary structure, but it can also adopt the tertiary structure. Why they can make the tertiary structure? Because right after the production of the secondary structure, it still have some space, it still have some of the feature to make another round of interactions in the regions where they cannot find any bond, where they previously do not find any bonds. So most of the times these tertiary structures are being found in case of tRNA, transfer RNAs, because these transfer RNAs have to have really strong and rigid, because these RNAs are acting like a transfer regions, or acting like an amino acid carrier. So what they are doing in this case, we are having the 3' end, which is slightly bit elongated, and we have a 5' end, which is short. Now this 3' end will bind to amino acid. Okay, and after binding to amino acid, this tRNA will come and bind with the RNA using the anticodon regions, with the code present in the anticodon regions with the mRNA, and finally it will this amino acid is being recognized and the protein synthesis will be carried out. Now this tertiary structure can be formed due to the interaction of bases, as we can find again the hydrogen bonding as well as the hydrogen stacking. Most of the part of this kind of interaction has been done using the hydrogen bonding. Now we can find UAU base triple in this kind of tertiary structure. Okay, so it is this kind of structure is done due to the presence of enormous rotational freedom in backbone. So not only they are having this rigid structure, they are having enormous flexibility to fold in right propagation, to fold properly, to make this kind of bonds. Okay, so thus it can fold into complex tertiary structures involving the formation of base triples and base backbone interactions as found in tRNAs. Okay, so in this picture we can see one of the examples of how they can further stretch the area of their bonding, or the freedom of their base interactions by attaching with 2 uracil. Now one adenine is there, this adenine can pair with 2 uracils to make another round of bonds. So forget about this uracil. So suppose these 2 adenine and uracil make bonds in normal situations to make a DNA secondary, an RNA secondary structure. So that is done. After the secondary structure, when the secondary structure came in and it finds some uracil residues right beside the adenine. Now this adenine will still try to attach with this uracil via the hydrogen bonding, and it actually survive to do that. It actually can do this, and it actually can establish this. So that the freedom for making bonds for the bases that we can find in RNA is much more improved, it's much more improved and improvised, as we can see in this picture. Okay, that's why the tertiary structure of the DNA is being formed. Now let's talk about some structural difference of RNA with DNA. So first talk about the chemical differences. As we know, in chemical differences of the ribose sugar and deoxyribose sugar is that the DNA contains the 2' deoxyribose sugar. In this 2' deoxyribose sugar contains only hydrogen instead of hydroxyl group in the ribose, and DNA also contains thymine instead of uracil. So that is another difference. Now what are the structural similarities, structural differences between DNA and RNA? That the secondary structure of RNA generally adopts A form of the helix, while the secondary structure of the DNA resembles the B form of the helix. Okay. Now another thing, that RNA has a much shorter range of nucleotides. Okay, it has a much shorter range of nucleotides and can adopt a widely variety of secondary and tertiary structure rather than DNAs. And why? That is really important point. And why? Because of the ability of the unusual base pairing that can be found in RNAs. That in RNA we can see this kind of guanine and uracil bonding. We can also see uracil adenine uracil bonding to make tertiary structures that you can find in our previous slide. Okay. So that's why in case of RNA, the degree of freedom to make hydrogen bonds are being improved. The degree of freedom to make base stacking interaction is being improved. That's why the RNA can be presented in various types of secondary and tertiary structures. Okay. Now if we start to talk about the structure of mRNA, then what we can find, the most of the mammalian cells, this mRNA is really very stable, because this mRNA is called the messenger RNA. Why? Because this mRNA is carrying the information of DNA from DNA and finally transfer it to make protein. So it actually plays a very important role for carrying the information for making proteins. Okay, and this messenger RNA is being made inside the nucleus. Then it is being carried out from nucleus into the cytoplasm via this ribonucleoprotein particles, as we know, and via those nuclear pore complexes. And when it comes into the cytoplasm, in the cytoplasm, all of this process of protein synthesis will go on. Now, if we look at the structure of mRNA, what we can find is various different regions are out there, and they are having all of those different regions are having different types of functions. Now, as you can see in this picture, this is the 5' end of the mRNA, and this is the 3' end of the mRNA. So in the 5' end, what we are having a cap. So cap means it's just like a cap that we wear. This cap is actually preventing the degradation of this 5' end of this mRNA. Okay, and what we are having a polynucleotide sequence like polyadenosine in the 3' end. So it is also blocking the 3' end informations. And why this kind of capping and poly A sequence is being tagged to mRNAs? Because this mRNA, when they present in cytosol, they can have vulnerable to damages because of the presence of different ribonucleases. Now this ribonucleases, if they start to cleave off from this region, it will take a long time to cleave this regions, or to cleave this adenosines before enter into this original codon sequences. Now why we take this kind of very measurements, previous measurements like this, prevention measures? Because in this RNA what we are having, the small version of the DNA codes. So RNA is actually representing the DNA, and if anything happens in the code of this mRNA, that will reflect in the production of the protein. And if a protein is produced malfunctioning or something like that, then it has been totally waste to produce those kind of protein inside the cell by using energies and all these things. So that's why we need to check this, that we can produce the right type of protein, or the right protein which is totally fully functioning. And also that's why we need to have a properly functional mRNA. And to have a properly functional mRNA, we must have a properly structured mRNA. So we can make this kind of structure. In this 5' end, we are having the capping. Now this capping is being made by the guanosine residue. It is being attached to this 5' region invertedly, and after the attachment invertedly, we can have this kind of strong capping, which actually prevents the hydrolysis process. Now this 5' portion and the 3' portion both attached with the UTR regions, and these UTR regions contain some stem loop structures like that. Now why this kind of stem loop structures are present in this this kind of 3' and 5' regions? Okay, this actually helps in the coding sequence, and these features in the coding sequence, as we can find, and this is also pretend to help this RNA to prevent those degradation processes. Okay, and we have a coding region in between. So coding region is totally separated via different regions like UTRs, and in those UTRs we are having this stem loop structures. If anything happens wrong in these places, or if any kind of bad protein enters in and start to chew up from both of the direction, whether 5' or 3' direction, then the stem loop structure is going to prevent that. First prevention measure is cap and poly A tail sequence. If they fail to react, then this stem loop structure start to block the activity of those enzymes, and finally if they are failed to work that, then we have then finally we have to reach the coding. So we are if some enzymes have to reach the coding region, or to block the coding region, or to degrade the coding region, it have to go through from this terminal portion towards the end of the coding. So coding is being put at the middle, or at the inside portion of this whole mRNA structure. Okay, and you can see also this in the 3 terminal portion, not terminal but slightly towards the coding portion, we have the AU stretch. Now the AU regions are thought to play a very important role in mRNA stability. Okay, now if we look at the mRNA of both prokaryotes and eukaryotes, we can find differences, that in prokaryote mRNAs, what happens, prokaryotic DNAs are having the coding sequences right after each other, so they do not having the junk regions that we can find inside the eukaryotic DNA, inside in eukaryotic RNA. So in the eukaryotic RNA is made up with several regions. Some of the regions are coding some proteins, and most of the part of it is unable to make any kind of protein. So all those regions which are not able to make any protein but still present are called the introns, and the regions which are capable to make proteins are called exons. So right after making the RNA from DNA, we do not usually call it the mRNA or proper mRNA. We call them the precursor of mRNA or pre-mRNA. Now this pre-mRNA have to have some modifications to finally make a functional mRNA. Now what are those modifications? Those modifications are the 5' capping, as well as the poly A tail arrangement at the 3' regions, and the secondary structure to make the stem loop structure in 5' and 3' regions, or something like that. Okay, but in case of prokaryotic RNAs, what we know that the prokaryotic RNA, when it is being transcribed from the DNA, we are having the stretches of sequences which are full of the coding regions. So the RNA is full of coding regions, so we do not need to cleave those introns from the exons, because there are no intron and exon present in this prokaryotic RNAs. So it is full of coding regions and full of regions that can make protein. Okay, that is capable for making proteins. Okay, so that is the thing. Now if we think about the capping here, you can look at the capping region like this. We have the 5' end, something like that. In here we have the triple phosphate group attached to it, and right after triple phosphate group we are having the guanine residue, and we have a sugar just totally invertedly attached to that. So if we look at the normal strand direction here, the 5', but in the sugar we are having totally opposite orientation direction, something like that. So it is placed totally in opposite orientation here. Now what it is doing here, it is actually protecting mRNA from 5' endonuclease attack, and increases the stability of mRNA in the initiation of the translation and in removal of introns. Now that's why it's important, because in case of the removal of introns, as we know, you have the mRNA. In case of eukaryotes, we have stretches of parts. So these are the exons, and again introns, exons, something like that. So what we have to do in this case, we need to cleave these introns out. So this part we have to cleave out. So how we can cleave out DNA and RNA backbone? We definitely need to use the need to use the endonuclease. Okay, so use endonuclease. Now this endonuclease is going to cleave this part, and then this intron part will be released, and only two exon parts are being attached with each other and to make structure like that. And these exon parts, arranged exon parts can make proteins, and these proteins are really important. Okay, so that's why we need to block both of the terminal portions for decaying them using the exonuclease, because in this process not only endonuclease are being released but also exonuclease are released. Okay, so that's why I need to block the 5' and 3', and we can block the 5' using this capping just by putting this nucleotide in opposite manner to this normal backbone of RNA, and we can make a cap in the 3' end by putting poly A sequences. Okay, now the histone mRNA lacks the poly A tail but have a sequence near the 3 terminal that can form a stem loop structure, and this also appears to provide the resistance to exonuclease activities that you can find in this picture. As you can see in the 3' end, this is the stem loop structure, and the presence of this stem loop structure, especially in case of what you can find in case of the histone mRNA, we can find this, and this part make the stem loop, and as a result of making the stem loop, it can prevent the activity of exonuclease. Okay, so it actually acts the substitute of the 3' poly A tail capping. Okay, now let us talk about the structures of tRNAs. Now the tRNAs are called the transfer RNAs. As we know, these tRNAs are actually the carrier for the amino acid sequences. It has an anticodon loop, and it also have the attachment site. Now the tRNAs is exactly looks like the characteristic L-shaped structure, and the tRNA is possessing the tertiary structure in between among those all RNA structures. Okay, and among all those RNAs, tRNA possesses various types of what we can say the modified bases like pseudouridine, ribothiamidine, or dihydrouridine, like this. Okay, so we have previously talked about a pseudouridine, where we have the structural difference in these regions. We have the ribothiamidine, and we can also have the dihydrouridine, where we having the 2 hydrogen groups extra present there in those 2 positions. Okay, so the structures of tRNAs, as we know, have to be varied from one to another. So there are different 20 amino acids that you can find inside our body. So there we must need 20 different types of tRNAs for carrying those different 20 types of amino acids, and that's what exactly happening. So we are not only having one type of tRNA, we are having 20 different varieties of tRNA which are specified for attaching with a particular amino acid, which are specified for carrying a particular amino acid and carry them into the protein synthesis machinery, proteasome, into the protein synthesis complex. Okay, now so in this case what we can look, due to the presence of this slightly modified bases, they can have huge enormous they further increase the ability of making the hydrogen bonds. The further increase the capability of making bonds between the bases, and that actually helps them to make really strong structures. Now if we look at the picture here like that, the interactions fold between these different loops. So this is the secondary structure of this tRNA. If you consider this is the primary structure, all of the primary structure will have this linear strand only having the backbone and bases just coming out from the backbone. Now in this picture, if it folds, and it folds something like that to make the C prime end just getting more stretched, and 5 prime is shortened, and here we have four loops. At the below we are having the anticodon loop. In this anticodon loop we have bases, and this bases having the complementarity sequence for the proper codon that we can find in mRNA, where this tRNA is going to bind. And we have D loop in one side and the T size C loop in exactly the opposite side. Now this T size C loop, we are having the pseudouridine present there. That's why we call it the T size C loop, because we are having the thymine and pseudouridine and cytosine right after one another. That's why we call the T size C loop. And the opposite side we call them the D loop. We are having the GC here. We are having the adenine nucleotides here, nucleotides, and they put together. So what is going on? This is the secondary structure which is pretty much stable, and we also have a short stretch of small loop which is called a variable loop, which is just in between the anticodon loop and T size C loop. Okay, now if we look at the structure, this is a structure that only can be made when we are having the stretches of nucleotide sequences that can have the freedom of making bonds between themselves more than the normal Arvind-Sharga pairing like AT and GC. So they can pair via GU pairing and all these types of pairings. And as a result of the presence of this pseudouridine and ribothymidine and dihydrouridine residues, they can further stretch this kind of capability to make another round of hydrogen bonding, another round of interaction, another round of hydrophobic stacking interaction between the flat faces of the bases to make a further round of complex structure, which we call a tertiary structure, as you can find in this picture. So this is the same picture which is denoting the which part of this region is start to interact with which part. So this G of this D loop is start to interact with this C, which is found in the variable loop. Now most of these interactions are being carried out from a D loop and T size C loop. So what we are generally looking at, just visualize this picture that this D loop is just coming out of this plane and it is going to interact with this T loop to make a folded structure, and this is the fold. This fold actually makes the structure the L-shaped and the characteristic L shape of this tRNA. Okay, so that's why this part is really important. We are finally folding this tRNA to make a tertiary structure. So everything is fine till that point. So due to the presence of this unusual bases, due to the presence of this modified nucleotide sequences, what they can do, they can pair bonds with themselves, they can pair hydrogen bonds with themselves, they can also make the hydrophobic stacking interactions between themselves, and finally they can fold. And most of the time this D loop and T size C loops start to interact. They will fold to give the characteristic L shape to the tRNA structure, and that can make a really stable structure. Now we can find the structure something like that. Okay, so this is the secondary structure. It will fold something like this. So we can have different loops like this. Just consider this is the D loop. So we are having this D loop here, and we are having this T size C loop here. So this T size C loop and D loops are interacting, as we can see. So the D loop, which have to be present here, so it start to fold in this orientation, in this direction, as you can see here, and after the folding this structure becomes a structure like this. Okay, Now at the end we are having the CCA tail, and this is the 3' end, and this is the 5' end, as you can see in this picture. Okay, so that's how this RNA structure is being made, and that's why the tertiary structure of RNA is being stabilized, because of not only the presence of the unusual type of base pairing, but also the presence of unusual bases. Okay, that's all, and I hope that's going to help you. Thank you.